Click the bell icon and subscribe to our channel so that you will not miss any classes.
Hello, welcome to class. In the previous class, we have discussed in detail about the structure of gynoecium and structure of pistil or structure of pistil and the structure of ovary and ovule. Now, let us discuss how the embryosac is developed in the ovule under the heading development of embryosac. Embryosac, since it carries female gamete, it is also referred as female gametophyte. Now, let us have a quick look at the structure of ovule as we discussed from the last class. The ovule is having integuments called outer integument and inner integument and the funicle, and inside it here there is development of embryosac and it is micropylarine and it is chalazoid. Now let us understand how this embryosac is developed. Before going to this, in a very young ovule, there is presence of a hypodermal cells which are actively dividing, or we can say them as meristematic, that is inside the integuments. And these hypodermal cells undergo differentiation into a special type of cells called archisporial cells. Again, these archisporial cells differentiate into two types of cells called primary parietal cells and sporogenous tissue. And from primary parietal cells there is development of integuments, and from sporogenous tissue there is development of embryosac. And remember, in each ovule normally there is development of a single embryosac, not more than that. And to start with the process of development, the sporogenous tissues which are present towards the micropylar end, one of it, that means sporogenous tissues are normally present towards the micropylar end. Among the many sporogenous tissue, one of it is capable of undergoing meiotic cell division, and that sporogenous cell is referred as, this is diploid cell, which is capable of undergoing meiosis. And this is what type of cell? Sporogenous cell, and it is normally present towards the micropyle. And this cell which is capable of undergoing meiotic cell division is referred as Megaspore mother cell, MMC. Megaspore mother cell. When this Megaspore mother cell undergo meiosis, we know that meiosis is a reductional cell division where the number of chromosomes reduces to half. Here is the ploidy is 2n. When it undergo meiotic cell division, produces 4 daughter cells which are having a haploid nature. So that is reduction in number of chromosomes will take place. As a result, 4 haploid cells are formed. And these individual cells are referred as megaspore. These are called megaspore, and initially these 4 megaspores are present together and they is referred as, and this structure is referred as megaspore tetrad. This structure is referred as megaspore tetrad. And this process, that is formation of haploid megaspore from diploid megaspore mother cell is referred as megasporogenesis. Megasporogenesis. Now, and we should have a track of the poles. So this is micropylar and this is chalazal end. Okay. Now, among the 4 megaspores which are present here, only one megaspore present towards the chalazal end will enlarge in its size. That means here this megaspore, I will write in different color. So this megaspore will enlarge in its size like this. And the remaining 3 which are present towards the micropyle will degenerate. So 3 of it will degenerate. So we can label it as degenerating megaspore. Degenerating megaspores these are, and the megaspore which is present towards chalaza, which enlarges in its size and later develops into embryosac, is referred as functional megaspore. That means it is functional megaspore. Next, the nucleus of functional megaspore, and you should remember here that the ploidy is N. The nucleus of functional megaspore will undergo 3 successive pre-nuclear divisions. That means the nucleus of it only divide but not the cytoplasm. That is the meaning of pre-nuclear division. Got it? So now here I will write here only. So the nucleus of the functional megaspore will undergo 3 successive pre-nuclear division, and among them first division I will tell now. When it undergo first nuclear division, forms 2 nucleus, 2 haploid nucleus, and 2 of them will move to the opposite poles. That is, one move towards micropyle and one move towards the chalaza. After reaching the successive poles, again they undergo 2 more division. I will write the continuation here. So again the single nucleus which are present at the opposite pole undergo 2 more divisions. So see here, it is 1 nucleus. When undergo first division produces 2 nucleus, and again 2 nucleus undergo 1 more division, form 4 nucleus. Now tell me, how many nucleus are formed at each pole? Yes, there is formation of 4 nucleus here and 4 nucleus here, like this. So this is 1, 2, 3 and 4 towards micropyle, and 4 nucleus towards chalaza. Remember, the nucleus are undergoing pre-nuclear divisions. Okay, so here 1 division and here 2 division. Totally 3 successive pre-nuclear divisions. After this, 1 nucleus from micropyle and 1 nucleus from chalaza will move towards the center. 1 nucleus from micropyle and 1 nucleus from chalaza will move to the center and stay at the center. And these 2 nucleus are referred as polar nuclei. These 2 are called as polar nuclei. And remaining 3 are present here. Let us look now what happens to these. And poles you should know here also, same is carried, it is micropyle and it is chalaza. Okay, and 2 nucleus which are present towards the micropyle will develops a cell wall. They get developed with a cell wall. They develops the cell wall, and they have special thickening these two cells which are formed by the formation of cell wall will have a special thickening called the filiform apparatus. Filiform apparatus. And these two cells which are formed by the formation of cell wall are called synergids. Okay, and you should remember that presence of filiform apparatus is a very peculiar character of synergids. No other cells in plant have filiform apparatus except synergids. Okay, and these filiform apparatus will play an important role during the entry of colon tube into embryosac. They secrete some enzymes or hormones or some chemicals which helps or which guide the entry of colon tube into the embryosac. Along with this, during the process of entry of colon tube into the embryosac, there is creation of some heat, and that heat also being absorbed by the filiform apparatus. That is the function of filiform apparatus. Okay, so we have discussed what happens to two nucleus. Two nucleus are covered with a cell wall and form synergids, and there is a special thickening called filiform apparatus. And the remaining one nucleus shares the cell wall of synergid. That means individually it is not developed with the cell wall, but it shares the cell wall with synergids, and that cell is called egg cell. Initially it won't have its own cell wall, it shares the cell wall with the synergids, but later during fertilization and all, it will develop its own cell wall. That is during and after fertilization. Till then it will share the cell wall with the cell wall of synergids. And this structure, that is two synergids and one egg cell present towards the micropylar end, is referred as egg apparatus. Is referred as egg apparatus. And coming to the chelazal end. Chelazal end also there is presence of three nucleus, and these three nucleus develops cell wall, and these cells, three cells which are formed at the chelazal end are referred as antipodals. And antipodals, they do not have any significant role. After fertilization, they will just degenerate. They won't do anything. Then, at the center, there are two polar nuclei present, and they also develop their own cell wall, but it forms a larger cell. How much large? I will show it now. From here to here. From here it is the cell wall. From here, this much cell wall it develops. So this entire yellow colored thing is one cell, okay, which is having two polar nuclei. And this cell which is present at the center is referred as central cell. Is referred as central cell. And the nucleus of central cell are called polar nuclei. You may ask now, even though they are present at center, we are calling them as polar. Pole means end, right? We are calling it as polar nuclei because they are from poles, from micropylar end and from chelazal end. Originally they are from the poles. That's why they are called as polar nuclei. And remember, these two polar nuclei will not fuse until the fertilization. Only during fertilization or after fertilization. Take my point. During, that is during the entry of colon tube into the embryosac, they undergo fusion, or it may take place after also in some plants. Okay, till then they are being separate. That is N and N. They are separate. Okay, now this is the complete structure of a embryo sac. When we look at embryo sac, there are 7 cells present, 2 synergids, 1 egg cell, total 3, 3 antipodals, total 6, and 1 central cell, total 7. And when we count the nucleus, 1, 2, 3, 4, 5, 6, 7, 8. Okay, so now completely developed embryo sac is 7 celled and 8 nucleated. 7 celled and 8 nucleated. And even though it is having 7 cells, it is developed from a single functional megaspore. It is developed from a single megaspore. Hence, this type of development of embryo sac is called monosporic development. This is called monosporic development. Monosporic development. Let us discuss some more types of embryo sacs, and this is the overview or total concept about the development of embryo sac. Let us take up now the types of embryo sac. Okay, there are 3 types of embryo sac. First one, as we discussed earlier, monosporic. Monosporic means it develops from single megaspore. Embryo sac which develops from single megaspore is referred as monosporic. Again, in monosporic there are 2 types. One is monosporic polygonum type. Monosporic polygonum type means embryo sac which is developed from a single megaspore having 7 cells and 8 nucleus is called monosporic polygonum type. And the second one is monosporic oinothera type. Embryo sac developed from a single megaspore but having only 2 synergids, 1 egg cell and 1 central cell. That is, it is having only 4 cells. 4 cells, and from 4 cells 5 nucleus. That means antipodals are absent. Antipodals absent. So such type of embryo sac is called monosporic oinothera type. And the second one is bisporic. Bisporic means, don't imagine like embryo sac developed from 2 megaspores. That is not the case. Embryo sac developed from a single megaspore having 2 nucleus, 2 haploid nucleus. Likewise, tetrasporic it is developed from a single megaspore having 4 nucleus, 4 haploid nucleus. So that is called tetrasporic. So these are the 3 types of embryo sacs. Now we have discussed in detail about the formation of male gametophyte and formation of female gametophyte. That means gametes are formed now. Male gamete is formed and female gamete is formed. Next is they have to fuse. But both of them, that is pollen grain and embryo sac, are non-motile. They are not having any organ for their motility. But they has to brought together To do so, some external agents or some process is involved, and that process of bringing the male gamete and female gamete together in plant is called pollination. Okay. From next class, we shall discuss in detail pollination, types of pollination and agents of pollination in detail. Okay.